and I see some new faces and old faces. So those on you, thanks and welcome for coming. And my name is Selina, and we are a couple of team. We'll get to them, all of us, at the end. So today we have a speaker, Mahendran Sithamparam. Yes, and who is actually visiting Japan, and he's a PhD student in astrobiology, and he's actually visiting my institute, Earth Life Science Institute in Tokyo, for 3 months. Yeah. And so today he's gonna speak about this really interesting hypothesis, which is quite old, but they have a new string in this hypothesis as well. And when I heard that when Mahendra's not doing science, he's actually playing football, and which is quite famous in Japan. And also they have been traveling, Mahendra and his partner, Pavitra, who's also a PhD student in Malaysia. You guys were traveling today and just came back from Osaka. And apparently Kyoto is their favorite place. Right, so Mahendra, thank you so much for doing this during your stay in Japan.
Hi everyone. Okay, my name is Mahendran, and Dr. Tilana gave me a good introduction about me, brief introduction. So I just jump straight to the topic today. So let me just break down the title of my talk. Okay. So let me just break down the title of the talk today. So a new way of Panspermia hypothesis. This is actually a proof of concept, because when we talk about space, we can't say that this actually happened, because there's no any written proof that, like our history, like a war history, whatever people write down, so we can relate back what happened. But according to space and everything, so what happened before the Earth formation and everything, nobody write it down. So because Earth wasn't formed, so yeah. Okay, so let's go back to the topic. Okay, Earth was formed 4.55 billion years old, but microbial life was only tested after 3.8 billion years ago. So what was happening between these two milestones? It's a big lot of time. It's like almost 1 billion years ago. I mean, it began. So what was happening between this period of time? So Earth was formed already. There's a planet. But how the life suddenly appeared from that particular planet. So that period of time is called as Hadean period. So if you know what is Hadean period means, just imagine that the Earth was like a fireball. So what we see in the movie 2012, so where the asteroid strikes the planet and everything, not the Earth is going down, but the asteroid strikes and then the Earth becomes like a fireball and everything. So that was like a Hadean period. So what was happening? You see, like Earth formed, Hadean period, and then suddenly life. So how do these things get relate to each other? So this is what I was talking. So that was like Hadean period. That's how our Earth looks like. So now it looks super beautiful from the sky and everything. But look at that old period of time. It looks like crazy and everything. From that era and suddenly to these beautiful animals full of flora, fauna, like us having this talk at British Cafe and everything, Space Cafe and everything. So how this happened? So that was going to talk. But I'm not going to give what happened exactly, but it's just a hypothesis. So before I go to the introduction of Panspermia hypothesis, let me give you the introduction. For the introduction itself, I'm going to give you. So let's start from the Big Bang nucleosynthesis. So Big Bang is like a famous thing for all of us because it's like a common word you can hear. So what is Big Bang nucleosynthesis? Nucleosynthesis is actually the synthesis of element. So we know that elements, like what we see in the periodic table. So any chemistry or any science field or whatever students or school students, whoever, they want to talk about chemistry, they go back to the periodic table. So now my question is that how the periodic table, the elements of periodic table was formed. So this is it. So let me just put it in a very, very simple term and explain to you how element was formed. Let's put it the most simplest element, which is helium. I mean, hydrogen is simplest, but let's go look at helium. So proton, after Big Bang, you know what's Big Bang, right? Big Bang is not just like an explosion. Some scientists say that's not explosion, but just the beginning of something. It's not just like a bang of explosion. So proton and neutron combine together, combine as fission. Okay, combine together, it forms another deuterium. And when another neutron combine to them, it's formed tritium. These two things, let me just get down here. These two things is an unstable thing. It's not an element yet, but unstable. But it can form, it can return back, it can decay back to the neutron and proton form. Or it can develop, it can more collision happen, it can develop to heavier and much more denser part. And when that happens, helium form. Okay? So when helium, again, more collision happened, and that is where heavier elements was being formed. So let's look at the timeline here. 13.6 billion years ago, in the first 3 minutes of the Big Bang, elements started to form. High temperature and density of the universe, and high energy and collision frequency, and electrostatic repulsion between the nuclei has been overcome, and then that first few elements was formed: hydrogen, helium, lithium, and so on. So this is exactly how the elements was formed. So let me show you in this simple diagram of Big Bang. So as you can see, that Big Bang happened after in the first 3 minutes the helium and hydrogen started to form, and not only that, along with lithium and beryllium. And after that, it's about how the planets are formed, how the stars are formed, how the solar system are formed. It's all after that. It doesn't happen in minutes, but as you can see, it's taking a million years, and then you see 13.7 giga years ago and everything. So I'm not going to touch on that later on. Okay. So I'm just focusing on how elements is formed right now. Okay, let me just show you this colorful picture here. So it's exactly the same thing, but it's more cooler picture. So as you can see on top, electron after the Big Bang begins, that's where time begins actually. And electron, neutron and proton started being formed, and then heavier elements being formed like helium and hydrogen after that, and helium atom, hydrogen atom, and then after that it's more to, you know, planet formation and everything. So this is actually a good way of seeing it. Okay, so this is the periodic table. This is actually an astrobiology kind of periodic table. So as you can see, from the Big Bang, hydrogen and helium being formed. Then after that, the later elements was formed when more collision, more combination, the fusion happens. Like for example, when large stars being formed, all the green colors are being formed as well. When supernova is actually an explosion of stars. So when that happens, and the rest of heavy elements are being formed as well. So I'm just focusing on the Big Bang nucleosynthesis. So there are other processes that gave us elements, like for example, stellar nucleosynthesis and photo disintegration that from gamma rays that makes that existing elements to decay and then form other elements as well. And radioactive decay of unstable elements, fission reaction, high energy particle acceleration, and nuclear bombs, nuclear reactors. So this is how the elements are being formed. As you can see, man-made. So this all doesn't form in the early, when the Big Bang happened. It's just formed after our life and the life now. I mean, us, the human. Okay, they just came up with this new invention, new elements over here. Okay, let's talk about how stars are being formed. So now I explained to you how the elements are being formed. Like for example, helium and hydrogen are being formed already, and that forms like a molecular cloud, and that cloud is called nebula. Okay, so that nebula is actually it's a dust, it's a cloud in the universe. Okay, so once that's being formed, and then accretion happens. Accretion means when the temperature and the pressure rises, and then the cloud starts to collapse. When that cloud starts to collapse, it forms like a stronger and in a round shape kind of thing. And then when clouds start to collapse, the smaller clouds also start to collapse. Okay, so and that's where protostars happen. So protostars can happen in two different ways. One is if the bigger cloud and collapse, it can form a massive star, or sun-like stars, like how we're having our stars right now. Our sun is sun-like stars, and that sun will will grow and become a red giant, and then planetary nebula, and then it can form to white dwarf, and then the cycle will keep on repeating. So now we know that we won't last forever unless we do something. So that is one sentence I want to highlight here. So that end of this presentation, I will read that back. And then another cycle is that it can form red supergiant, and then it started to explode. Supernova is an explosion, and then it can form a black hole if it's too big, and then or a neutron star, or cycle back because its explosion, the dust will be still there, and then the cycle will keep on continuing just like that. Next, I want to talk about how planetary bodies are being formed, like how the solar system, how planets and everything are being formed. It's the same process actually. So it's accretion, initial growth in the protoplanet core, dust and ice particles together in growing clumps. And then when the clouds collapse, it also, as I said, that it's formed different layers, different region of clouds are being formed as well. So planets get layered internal region when due to difference in density of matter. So even the cloud, the inner one will be there's a different density. That's the reason we have the core of a planet, core of a sun, and everything. Okay, so now let's fast forward to how. So what are we made up? So let's look at this. These are I talk about how planets are being formed. I talk about how elements are being formed. So now let's talk about what are we made up. So I'm just showing you is the DNA structure here. As you can see, this DNA, this simple DNA structure is our building blocks are actually CHNOPS, which is carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So these are the most fundamental and the building blocks of life as we know. Okay, so from this periodic table, we know how this one is being formed. We know what is what we are made of. So, like for example, scientists have found out that how elements are being formed. Another set of scientists found out that what we are made of. So now the linkage, elements to life. So how that happens, that is the one we don't know. How from element and suddenly become life. So we know the life building block, but we don't know what happened, what the magical thing happened that suddenly the elements join together become a life. So that is the questions of astrobiology. So that is why the field of astrobiology will try to work on. Astrobiology is actually the study of origin of life on Earth, and also the search for extraterrestrial life, and to see how the formation of life, what's the life on other planets, how their habitability and everything. So astrobiology studies about the original nature of the life or evolution of planet, habitability of, for example, now the very famous things is the habitability of early Mars, and then icy world, Europa and I think Europa is the one famous that the GWS and everything is working on, and solar system and beyond solar system, like exoplanets, like what our Kepler systems we are finding out about the Kepler systems and everything. Okay, so what is astrobiologists currently doing is that I would say that it's very difficult to define life. So why is that? We don't know second known life. We only know life on Earth, life as we know it. So we don't know what is the alternative life. We don't have alien life on other planets, or the life I'm saying just not like a human or big structure or alien flying with a robot or whatever. It's not that, but just like a small microorganism that we can found in Mars or whatever. So we don't have that evidence. So we can't compare what is life on Earth and another life. So we can't really define it. So that is the problem about life. Okay, we can't compare to another life, and then we don't know, is this how we can make life, or is there another alternative method for it? Okay, the questions that to answer this, so there are several famous, big areas that astrobiologists will be working, which is, are there life on other worlds? For example, this is our solar system, okay, and this is the Kepler system. So Kepler system is exoplanet system. So they see that some of the planets are Earth-like planets, like you can see this photo here. It's a bit similar to Earth, but we don't know what is habitable. Life is that life there, or is it some kind of movement or anything about life? We don't know that yet. So this another big questions that astrobiologists will be working. And other origin of life, for example, the life I'm talking about, like I said, that it does not have necessarily have to be like our life. It can be in like a different kind of robotics, mosquito, or this kind of big whale walking on the land, or I don't know what is this, like a dinosaur having flower head, and this kind of turtle, giant turtle. It can be like this. It can be like whichever. It's just all artist interpretation. So we don't know whether this one really exists out there or not, because the universe is so big. So if you ask personally, I personally believe that there are life out there, but you just don't know to find out. It's beyond our capability to reach that. So the limitation here is that we know that we only study about our solar system. Even about our solar system also we don't know much, because Europa is icy world. So now we are interested in that to find what's inside the icy world, what's happening and everything. So we only focus on our solar system. What about the rest of the solar system? Maybe our neighbor solar system or whatever. So our limitations there. We are only focusing there. Another thing is that this is our tree of life. So this is our flora and fauna for now, the bacteria, eukarya and archaea. So when we come down to these, we can go for last universal common ancestor, which is Luca. So how this Luca is started, we don't know that. So from non-living things, the periodic table, from non-living things to living things, how that happened, we don't know. So let me just show you a bit of chemistry here. And as I said that Luca, scientists debates in different world. Some scientists say that RNA world, scientists from RNA world, they defend themselves that RNA formed first. Another set of scientists say that for RNA to build up the RNA, it has to be metabolism first. And what if then the membrane will come and fail that? Okay, you got the RNA and everything. So where is the cell going to be? So membrane, you need a house for that. So membrane will detect that this was formed first, and then the clay world, and then the thermal vent world. Everything keep on arguing that which has happened first, but we don't know the answer. So that is why this is remain the big question. How does last universal common ancestor begin? That is the big question here. Okay, this is an interesting picture. Just put it here. So if you can see that the picture number one is the picture number one is starting from the very simple elements, which is carbohydrates, nucleus. Like before this, I'm talking about the periodic table. Okay, so after that, it's like a big jump. We are starting to talking about the smaller molecules, and after that, it's formed to a bigger complex molecule. Okay, so it's like a developing and evolving, and then from there it's formed the world they are talking about, like a protein world, RNA world, and there will be a membrane compartment world. And then from there are some further evolution happens, and replication, metabolism world, and then now after that it's like a cell, like a very simple cell, protein, RNA, DNA, life, like a very simple cell is being formed. So this like a flow like that, but we don't know whether this actually happened or not. We can't really confirm this, because as I said, we can't define life. It can be from one and then say to here, and then it break down to here, and then it can form back. We don't know the correct mechanism or pathway yet. For that, we have to know origin of life on Earth first before we going to find other planets, other life or whatever life on alien life. So we have to know what's happening to us in our Earth. So there, as you can see, there are a lot of pathways here. It's like how life can begin. Like you see, like raw materials. We are talking about the raw materials. What are the raw materials for life? As I said that this is a CHNOPH. We know that life is made up from that. But from where we are getting that? Is it that the elements already delivered, or it's already on Earth for it to just happen just like that, or it's coming from another place, or it's what happened? So that is what they're talking about. Prebiotic organic compound delivered from elsewhere from solar system, including carbohydrates, chondrite, meteorite, comets, and also chemical synthesis on Earth. So it especially exclusively happened on Earth. It was formed on Earth. So all these are possibilities. We can't say that it's happened. So to understand that, these are the fundamental questions of astrobiology. The astrobiologists will be working on this mind map here. Like for example, you see, does life exist elsewhere in the universe? So that's the question. Is yes or no? It's remain debatable. So if it's like it's an ongoing exploration. Like we send the JWST, we are going to now the JWST giving us a lot of good information, like very interesting information. But that is limited to our neighboring place. So what is happening in beyond that? So we still don't know. So that's always going to be ongoing process. Even we found another life, we found our friend alien, it's still going to be an ongoing process. What's happening after that and beyond that? So like we focus on our solar system, includes Mars and Europa. These are the very famous ones, like Elon Musk trying to colonize Mars and how it's going to go on. And Europa is an icy world. What's happening under the deep ocean? Is it some kind of fish swimming there, or we don't know? Could be, but we don't know. So just that beyond our capabilities. So it's going to be ongoing process. Maybe one day we can find out what's happening all that. But for now, my interest will be in origin of biological important molecules, including interstellar cloud that I told you just now, and comets and asteroids, and planetary atmosphere, surface, and interior that can give us the organic compound. Sorry. Okay, so the one I said earlier, so this part, including the elements of life was transported by comets and asteroids, could be like this. All right.
Just
Okay.
blocking.
This side. Okay. So this could be a visual that I can show you, like Earth. This Earth. Okay. Okay, the Earth's not going to be like this. It's like already forms like a habitable Earth. But just imagine it's like a old Earth, like a fireball Earth. Comets are bringing in elements that was formed elsewhere. Like for example, maybe this particular meteorite could have brought the basic elements of life all together, or just one of it, maybe carbon only. This meteorite is fully rich in carbon, or maybe another meteorite fully rich in sulfur. It can bring it from elsewhere, it's formed elsewhere in the universe, bring it to the Earth. So that is the hypothesis is all about. That's what is Panspermia hypothesis. Panspermia hypothesis suggests that life on Earth did not originate from Earth, but it was brought or the building blocks was brought from elsewhere through meteorites or comets to the early Earth. So this is actually a very old hypothesis which was was mentioned by Anaxagoras, and after several centuries was shown interest by Swan Arrhenius, and it's still going on. Nobody has found out that. So you can imagine how big is that thing. We can't really find the answer, even it's taking so many years. So as you can see in this picture, maybe the comments here doesn't bring the elements of life, but some kind of microbial life from elsewhere itself. It can happen, we can't say yes or no. It can bring to this fireball kind of earth, the life is already formed and bring it to the fireball earth, and that fireball disintegrate the element, and then another tree of life started to grow from there. Like life already there, it's bringing out to the earth, and then it's disintegrate to basic elements, and then from there it forms a different pathway to form a different life. It can also happen, we don't know. So that was a very big study of Panspermia hypothesis. Okay, the fireball was talking here. What was it? It's actually late heavy bombardment. So late heavy bombardment happened almost like 4.1 billion years ago. So this is why this hypothesis is very interesting, because we can relate that to this. So we have found out that Allen Hill's meteorite, which was found in 1984, it contains a lot of chemicals, macromolecules and everything. So what I'm trying to say is that these are not from Earth, it's from elsewhere. So that means this is already elsewhere in the universe. So you see, you can if you can see the combination that something happened outside of the world is brought to the Earth. So is that late heavy bombardment dust from a lot of meteoritic impact happened from elsewhere, it brought to the Earth, and then that is the reason we have our carbon, hydrogen, oxygen, all the building blocks of life on Earth. It can facilitate that flow of origin of life. That remains a question. So what is late heavy? How this late heavy bombardment happened? Because of this migration of giant planets. So this diagram here shows that our initial solar system, where as you can see Sun in the middle, and then the first planet is Jupiter. So this is nice model suggests, nice model is not, I'm telling that it's a very nice model, but it's actually, the name was given to this model is actually nice model. So they suggest that Jupiter was the first planet. After that, it started migrate for whatever reason, and it's going to disturb the outer, smaller comets and meteorites, which was talking when I was talking about the formation of Earth. When the nebula collapsed and then the smaller outer clouds to collapse and forms a smaller body, planetary bodies. So that could be this. Meteorites, a lot of meteorites are being formed at the outside. So these big giant planets go and disturb this, and then that started to move all around direction. So that is the reason we get late heavy bombardment. Not only Earth, but the neighboring planet as well. For example, this can happen. This piece of chunk is actually this small. This is actually a meteorite, Martian meteorite. So these Martian meteorites from Mars. How from Mars came to Earth? So maybe a meteorite going hit Mars. A small chunk of Mars was released, and then that floats or maybe fly towards Earth and land on Earth. So that can happen as well. So this is the whole scenario of Panspermia hypothesis. Okay, this hypothesis is very interesting. So that's the reason JAXA has initiated a mission called Tanpopo mission. No, it's going to be behind supposedly. Okay. I was waiting for this graphics. That's why I was, Okay, so back to the topic. So this Sithamparam mission is actually to test the Panspermia hypothesis. It's actually to expose the sample at outer space to see their survivability. So how this life can survive at outer space on where we need oxygen to breathe and our life needs to some kind of do in an earthly environment. But how does this small bacteria or whatever microorganism that's in outer space, can they survive? Can they relate to what we are talking about, Panspermia hypothesis? Panspermia hypothesis have to travel through the space, enduring all the space condition, the cosmic ray, high temperature, and then very low temperature as well. It has to withstand all these parameters. So that's the reason they want to test it at low Earth orbit first, using the Kibo module. This is the Kibo module I was talking. So this is the exposure panel here. So they will expose some kind of samples. It can be microorganism or any kind of samples. They will expose here and then they will analyze it. What's the degradation, what it does something magical, or does it die immediately? So what is happening? So this is actually to test the Panspermia hypothesis. So what? Okay, so let me just show you one interesting results from Sithamparam mission. So recently they exposed a bacteria called Deinococcus species, and for 3 years. So what they noticed that after 1 year it started to degrade. So that means in the first year it was surviving at outer space. So that means there are certain things can survive at outer space. And what is sad thing is that it only can survive in one year. After that it started to degrade. And once they brought back to Earth, it's repaired their damaged DNA. So this is very interesting. So that means it's died halfway and then brought to Earth and started to release back. So this is very amazing. But what am I thinking is that, what if rather than be exposing Deinococcus species, like for the bacteria or complex molecules alive, why not we just expose non-living things, or maybe a non-organism based seed, Panspermia seed. So let's check what happens when we just expose a simple structure, simple non-organism stuff. So what am I talking about here? So why not? Okay. Before that, I'm just going to show you here. This is actually a type of Panspermia. We are talking about the meteorites and everything. So there are other things as well, like solar radiation and free-floating elements from outer space, and also directed Panspermia is like advanced civilization. They are purposely seed our life. They are too advanced. They will feel very boring. You just want to just throw something on our Earth. It can happen like that. This sounds very crazy, but that's what we are trying to do. We are trying to do at that advance. Okay, as an extension of this Panspermia hypothesis, we introduced material-based Panspermia hypothesis, which is actually exposing non-organism to outer space. So we recently published a paper on this, and then here we explain how it can happen, why it can happen, what are the things, what are the material-based that we can expose at outer space. So what will be the material Panspermia seed? My answer will be polyester gel. So let me just show you what is this polyester gel is made of, what it's going to do and everything. So initial alpha hydroxy acid. So alpha hydroxy acid under a simple wet drying condition. After drying condition, we just took 5 types of alpha hydroxy acid. So we just mix it together and we dry it for 80 degrees Celsius for 1 week. So what happens to that solution? It can form a gel state. It's like a gel. It's a transparent gel. So another interesting thing that gel can do is that after re-wet it using 4:1 ratio of water and astronitrile, it can form membranous droplet. So what is this membranous droplets actually? It's formed by liquid-liquid phase separation. So what is interesting thing is that it can form like chemicals, something drying happen, and then it can form like a housing area. It can form like it can absorb material outside of that droplet, bring it inside, and then it can release back to the outside. It can do something interesting here. So that was also tested by using RNA tag, fluorescent tag RNA. It can absorb from outside towards inside. So that is the reason we want to use the polyester gel as non-organism based seed and expose it to outer space and see what happens. So what are the things that the membranous droplet can do is that it can release and absorb RNA, it can fold proteins inside, and it can accumulate lipid around it. So that means from membranous droplet, it can form membrane bound droplet. So this wet drying cycle, if you can see this drying, wetting, or wetting, drying, the cycle, we can relate that to rain and evaporation, or high and low tide on early Earth, or maybe big wave, as we can see in our movies, tsunami and whatever. So it can happen like that, like where Earth was already formed, hydrothermal vent. We can relate this to a lot of world I was showing just now. Where if there is rain, atmospheric rain, that's where the alpha hydroxy acid got wetted, and then daytime sunlight, it can dry it, and then just get formed, and then it can happen by its own, like a natural environment. So why alpha hydroxy acid? So why we simply want to use, because we can't really simply take, okay, this chemical is good, just want to use it. So we have to relate back to a special thing. So this alpha hydroxy acid is a prebiotically available organic compound. So a lot of study has proven that the meteorite, the comets I was talking, there are several comets have proven that alpha hydroxy acid, a lot of alpha hydroxy acid is being contained in that meteorite. Not only that, the famous Miller-Urey experiment, the spark discharge, also formed alpha hydroxy acid. So what is happening is that these things was brought to the early Earth, which is prebiotically available. So that is the reason we want to use alpha hydroxy acid to synthesize the gel. So it resembles that it's happening by its own, something like that. Okay, so this graphic, what's my project going to be? So this is the donor planet here. Donor planets. So the CPG is actually combinatorial polymer gels, which is the polyester gel. So it has to endure the space exposure and then go to recipient planet, and then once it touches the water body, it can do the magical stuff where liquid phase separation happens. It can absorb materials inside and release. So we know this thing can happen. We know this thing can happen, but what we don't know is that whether it can sustain and remain functionable after all these space travel. So why polymeric gel can be the Panspermia seed? So basically it can do these four things, which is material exchange, motion, coalescence, and evolution. So I will go through this one by one. First thing is material exchange. So here we can see in three ways. One thing is that polyester gel can bring in pre-encapsulated material. So that means we can put in some chemicals or molecules inside the gel and then transfer it to another place, let that do their spontaneous activity. So that is one way of saying it. Another way is that the gel can go to the new place and then form the membranous droplet. That membranous droplets can facilitate chemical exchange in the new environment. Or the third one is that gel can go to a new place, disintegrate. The gel is made of alpha hydroxy acid, go to the new place and disintegrate that and release, settle down the new chemicals over there. Maybe some kind of spontaneous activity can happen, as what I was talking about, the Panspermia hypothesis, were transporting elements from one place to another. Second thing is motion. So this membranous droplet can do movement. So this motion is important for bringing one chemical to another. So it's absorb one chemical, certain chemical is good to move to another place and release that, or maybe intake another set of chemical and can do some kind of spontaneous activity inside these membranous droplet. So this is actually a microscopic image. Microscopic image. That why it's not moving. Yeah, it's actually a time lapse. It moves super slow. So that means it can move from one place to another while absorbing. So this depends also on density. So the more the droplets, the faster it moves, or it's moved in one direction because of collision and everything. Next is coalescence. Coalescence is when few membranous droplets combine to each other and form a bigger droplets. So what's happening here is that this membranous droplet, okay, I spoiled it. It's okay. Okay. Why is moving automatically? Okay, so this membranous droplet is have a round morphology where one and each other come into contact, it can form a bigger droplets. So why this is important? Because when it uptake the chemicals, it has to prevent from extinction. So it has to provide a room for bigger area for chemical activities to happen. So basically this is actually like a formation of a cell itself, a protocell itself. So a membranous droplet taking in chemical, it has to do some kind of activity inside the droplet. So for that, it need to have a sufficient area for the materials to come in and do some activity. So that is why this coalescence is important. So as I showed you just now, droplets strong together are strong. So like this moving. So when it's small enough, it doesn't give enough area and it will go extinct. It will just die just like that. It stop just there. Next is evolution. Evolution where membraneless droplets become membrane-bound droplet. So where it can lipid scaffolding can happen around it. From it's like a droplet, and then it's like a membranous droplet from liquid phase separation, and then it forms lipid structure outside it. So it forms membrane. So that is a very interesting thing. Okay, here I'm showing our modern biological cell. So this is actually very complex cell. So what I'm trying to say is that rather than focusing how this cell was formed, how building blocks of this cell, like when we flip our textbook, what's our life is made of, building block of life is cell. So what is the building of cell? So we don't want to focus on that. Rather than that, why don't we focus on very simple molecule instead? So like this. Just these simple membraneless droplets over here. We just focus on here how it uptakes the chemical, release the chemical, how it's from here it developed through a bigger structure, and perhaps these smaller ones can become like a bigger cell when it's taking enough chemical to do some activities, spontaneous activities. So for that, for all these things to happen, these are the questions that I need to answer. Firstly, what are the kind of CPG, what are the kind of polymer gels that can be synthesized by using a biotic process? So earlier I showed you only 5 types. So there are many types of alpha hydroxy acid. So I need to do some kind of screening to choose the best things from that. Another thing is that what are the level of degradation that gel can degrade and its robustness towards the simulated space exposure. Lastly, how does if this integrate, if the gel degrade, how does that affect on its functionality? So that means the 4 functionality I was explaining just now, does it allows it to do the 4 amazing thing or just make it to stop everything? So that also I have to test. So how am I going to do that? So firstly, to synthesize the CPG, I have to filter out all the monomers, and then after a drying cycle, it can form the CPG, and then I have to do some instrumentation analysis using mass spectrometry to see what is the specialty of each combination, whether which combination forms a better one, which combination can form a longer chain of polyester. All these kind of things can be do in the first place. Next is radiation radiation exposure, and on this space exposing stuff. So I'm going to speak the first, the basic easy 5 parameters, which is UV radiation, temperature fluctuation, microgravity, gamma radiation, desiccation. So I want to test how this polyester gel can survive all these outer space condition. So So this will be the ground-based experiment rather than before bringing it to outer space, as I showed in Tanpopo mission. So we have to select the best candidate. For that, we have to do the ground-based. So all these experiments will be ground-based laboratory experiments by using simulated instrument. And then after exposing it, I have to check whether it can form the membranous droplet, and that membranous droplet can do the functionalities I was talking earlier or not. So if everything goes well, if let's say I have answered, before that I have to show these pictures. These are the pictures I've took recently. This is the combination of all 5 chemicals over here. It can form this membranous droplet. If I use GA only, it doesn't form a droplet. It forms some kind of crystal structure of out of shape structure. But other than that, everything forms the droplets. So here we can see that, you see, if GA by itself doesn't form a gel, but in combination with others it forms, GA by itself doesn't form a droplet, but in combination with others it forms a droplet. So this is actually a big topic for me to filter out. So definitely I can't do it everything, but I'm just going to pick the easy ones as well. Okay, this is actually a multi-mass spectrometry data, so I don't want to go too technical here. I'm just going to show that long polyester chain can be formed by using this chemical. So why this is important? Because it shows whether the chemicals forms the gel or not. So that is why we have to do all these things. Another thing I want to tell is that based on that screening process itself, there's a different things that can be played as well, which is different temperature. So just now I was talking about 80 degrees Celsius. So what if we increase or decrease the temperature? Does it form the droplet or not? This is also a thing that need to be tested. The paper's already been published. So here it's been found out that shorter temperature doesn't form the droplet, but lower temperature doesn't form the droplet, but it also forms a shorter polymer. But higher temperature doesn't forms a droplet, but there's no big changes as well. So 80 degrees Celsius forms the gels and droplets, and after that it doesn't give a bigger difference. So this is actually a big thing to find out. So what if, because we know that outer space does not have 150 degrees Celsius, maybe hotter than that, higher than that, or very much higher than that, or very much lower than that. So we have to test all that. So next is chirality as well. Chirality means like the type of chemical, like mirror image of chemical. So what we use, L-lactic acid, D-lactic acid, is a different type. Even lactic acid itself has a different type of lactic acid. So how does that play the role in formation of gel and formation of droplets? That itself has to be found out. So what I'm trying to say is that on the first question itself, there are a lot of things has to be done. So rather than exploring everything, just cut short everything. I'm going to cut short a few things and then go to the second question. So let's say I have listed down the best combination, and then I have known the level of degradation, and after the degradation it remains functionable. So that means, can I send that to outer space already? So does it, my project going to be successful? Definitely not, because we can relate that to this picture. We can think that our work is going to be almost done, and then once we are done the last phase, the last thing, then only we see the big mountain after that. Similar to my project, I have a lot of questions that I myself have to be answered. So first one, I'm just going to go through the very, very simple ones. Like for example, I was testing the space conditions separately, but outer space happened everything together. So how does that going to affect the gel? It's like you are fighting one
bodybuilders each time. So what if all the 5 bodybuilders come to you, attack you? So are you going to live or die straight away? So that's what we're going to test with this gel. So I don't know about that. How about the thickness of that? Thickness of the gel plays a role. Is it a bigger gel? I mean, a big volume of gel can survive, or a small little gel can do the things that similar to the big amount, or the cosmic radiation I'm only seeing on 5 types of environment and outer space. So there are other parameters as well. So I haven't tested that. So these are the things that a lot of things has to be done, because what as I said that this is actually a big topic, so I can't answer everything. If I want to do everything, it's going to take a longer time. So what I'm trying to do is that before I think about this, this is my motivation for my project, where we don't have to see the whole staircase, we just take the first step and then see what happens next. And then we found out that, and then I can further develop. Back to this. Okay, so for example, I'm not sure, I don't know whether the gel can survive or not. So I have to do the exposure first. If let's say a gel can survive a certain level of it can degrade to a certain level. So what the combination I can do? For example, maybe I can play the ratio rather than putting everything equally divide, and then I can put, for example, I take 80% of lactic acid and then I add on with 2% of 20% of phenylic acid. Maybe that can be bringing a good combination. So if before planning everything, I just want to jump into the ocean and then see what happens one after another. So that is what I'm going to do. So finally, a take-home message for all of us. So we cannot see life on other planets by using our present-day biology. So we need a material-based Panspermia seed to do at least a comparative work with organisms. What can organisms do in outer space? And we can compare that with a non-organism, what can do in outer space. And finally, as I say that, once we know that, further experiment can begin with this starting point. Perhaps they can take this gel and apply it to other further experiments as well. So before I end our talk, so I just want to show this amazing thing that was mentioned by Stephen Hawking. He said that we humans will not exist or if we doesn't go out of our home planet. So for this to happen, we don't need to know whether a planet is habitable, or we as a life, as human ourselves, take a spaceship, space shuttle, go to another planet and start to do our everyday life immediately. So we don't know that. I mean, we no need to do that. So all I'm telling that we have to know whether a planet can facilitate a water body can. A planet that has a liquid can facilitate chemical activities or not. If that can happen, then maybe this Panspermia hypothesis, by using these polyesters, can do the things starting from a cell, as what happened in the early Earth environment. So lastly, I want to give my acknowledgement to the team, and I'm currently working under Dr. Tony and in his lab. So all the polyester world and polyester findings was all done by Dr. Tony and his team, and I'm carrying that invention, that findings, and going to apply that in Panspermia hypothesis. So my project is still going on. So perhaps in maybe 1 year time or few months time, I can get some solid evidence to show whether it's doable and it gives a positive feedback, positive results or not. So that's all from me, and I'm happy to take the questions. If you don't understand anything, just shoot out the questions to me.
Thank you. Thank you so much, Mahendran, for very in-depth and interesting theory sharing with us, and your work. I think there's more questions than answers
Yeah.
to begin with. Right. questions. Let's start with Chase.
Hi, great speech. Not so much a question, but something that I've read up on in the past that may do well with what you're testing. So two things that I've read on is, number one, I want to suggest fungi.
Okay.
They do not use the same rules of life as floral or fauna, since they don't photosynthesis. They can live as spores, and they can live without oxygen for a very long time. So it's just something to look into. As well as, are you familiar with tardigrades?
Yeah, that is they can withstand high temperature and everything.
And I read an article that apparently they survived in the vacuum of space and came back to life when they brought it in.
It's a very recent findings, I would say. That, yeah.
Yeah, tardigrades are like the most toughest creature on Earth.
Thermophiles can withstand high temperature.
Yeah.
So that my topic is then we are focusing on that. Tardigrades and everything is a complex molecules. It's made of, it's alive already, right? So I just want to test, what if we send an empty, let's say tardigrade, but an empty tardigrade. Inside tardigrade is nothing, just a casing of tardigrade. So what's happening to that thing? So just start from basic. After that we can develop from there. So that's my idea. But that your words are make sense also.
Yeah, and tardigrades are microorganisms, so they fall into your category of what you're testing. So, but the fungi, I just think it would be an interesting one because they are life, they're not floral, and they don't have the same rules. So.
Okay, maybe I should try to read that to the topic.
But if you watch Last of Us, don't mess with fungi.
That's right. The world's largest organism is actually a 50,000 mile fungus organism that spreads throughout an entire continent underground.
Anyone else? Let's go from the back.
So, just out of curiosity, I guess, for the panspermia theory in general. So maybe your take about that. So there seems to be well, my understanding at least, you can correct me if I'm wrong, there seems to be necessity of something like traveling. It could be materials, as you're studying some kind of polymer. It could be molecules. It could be say some super alien, just dropping life droplets, whatever they could be or whatever. So behind this panspermia, are we saying that there are some conditions that are missing for this specific, say, part of the universe where the Earth is, so we need help from outside? Or are we saying that there is something troubling and that's just speeding up the process?
In my opinion, I would say that we don't know the pathway yet. Let me repeat your question. You're saying that is there some kind of catalysis that happened to make the life to happen? Is that your question?
Yeah, in a way, it seems the whole idea seems to be, okay, there's something that traveled, it could be it could be coming from meteors, it could be coming from comets or whatever. And the question is, why are we looking into that? Is it because we think that these, say, these objects actually brought some special, say, materials, some special molecules, some special something that was missing, to create life? Or why do we need it?
Oh, okay, that's a very interesting question because, Earth, when Earth forms, maybe it's already contain all these chemicals. So, what is there will be some kind. What I'm trying to say is that, although it contains several things, maybe the important part was not there. So, for example, there is a paper by a scientist. He say that life. When we are talking about life, we say that proteins, amino acids can come to proteins and forms RNA and DNA and everything. So another paper said that alpha hydroxy acid, it's make it like a catalyst. It comes in, do some work, and then goes out. It doesn't involve in the making of the life. So that can happen. For example, as you said that this CHNOPS is important to come in and build the life. But what I'm trying to say is that that rather than focusing on DNA, so why not we focus on the simple stuff? What are the casing, the surrounding of that cell? So that itself can be made of CHNOPS. So that is what I was trying to bring into here. Yeah.
Let's go here.
Well, I have two questions which sound a bit different, but they're actually part of the same theme.
Okay.
First of all, so if I understood correctly, your reasoning to choosing that specific material is limited in the way we understand what happened on Earth, in the like process of formation of Earth. However, if the formation of another star was different, like of another planet or where that life originates is different, why are we restricting ourselves to the chemicals which are like the combinations which are arose by the Earth-based phenomena? So because like the reason you're choosing like this type of gels, if I assume correctly, is because on Earth there was a certain history of formation.
Yeah.
And the second question, which was kind of part of it, is if you were to design a system from scratch to travel, like to transfer life to another planet in the way we know it, which system would you choose? Which, if you're unrestricted by the idea that it's like it has to be the same or natural, like we can create it a lot like from scratch.
Okay, so to answer your first question, so before we try to look at other solar system, so like how the life, or maybe if there is some kind of life, so how it is formed, maybe the life on that particular different solar system, like for example we breathe in oxygen, just put in a simple context, so breathe in oxygen, perhaps that life can breathe in some kind of poisonous gas for them to live. So rather than focusing on that, we have to know how our own life was originated.
No, but if our life is brought from another planet originally, as you say, and you will want to know how it was reacting to the space travel, right?
Correct.
So right now you're testing it by using the life which could have arose on Earth and how that will travel. Like you understand the contradiction which I'm talking about.
So let me repeat your question. You see that
Sorry.
life coming from, life from outside,
So
elsewhere?
So the theory is that life on Earth originates from somewhere else. Like if it was brought by meteorite or something. That's the
Okay,
theory.
so Panspermia hypothesis, that's not only life, but the building blocks of life.
Building. Perfect. That's actually right. So, however, you're testing the building blocks in space based on those which could have been produced from Earth originally. If I understood correctly. That's what did click for me. I'm sorry if that's.
So now the limitation I'm putting in my project is that I'm not looking into the chemicals that already in the Earth was formed. So I'm looking from, if let's say that important chemical was brought from elsewhere. So what happens when that thing occurs?
So you're basing it on what would be necessary to begin with on Earth.
Correct.
Okay. So you're already designing it, not thinking about what. Okay, so if you were designing from scratch and not thinking about anything on Earth life in general, what would be the best system to transfer this, so which kind of system which will self-replicate and maintain its own entropy, which will be sent between Earth, which is not a robot, what would be the best system for that?
So your question is that, so let's say we know our recipe of origin of life, so you're telling that which system we can send.
So for example, we can create this, because in synthetic biology there is like gel-based life already, which can self-replicate and maintain its entropy by. So would we go for gels or would we go for something else, for another material, for this sort of transport?
For the transport, is it?
Yes.
For the transport, it's like, that's why we want to know how the degradation happens out of space. If the gel can degrade, okay, so that means we can encapsulate the gel itself by using some kind of rocky structure. So for example, we can take, we can encapsulate the gel itself by a meteorite compound, like maybe rock. So send it out to space and see how that rock, that layer of rock can protect the gel. If that can happen, maybe that the space travel can we have to we have to start from somewhere. So that is why I want to expose the gel itself to outer space and see how the degradation happens.
Okay.
Yeah.
Okay, thank you very much. I'm sorry for
No problem, thank you.
Let's go here and then the last question.
So, one question. You were apparently telling that you were experimenting on chirality of all those molecules, about how they were eventually building membranes or not building membranes. And on Earth, Earth's life has specific chirality. If I'm not mistaken, it's the right-handed type chirality for the molecules used in life. Did you saw some correlation in your results in the ability of those gels to form structure with that right-handed chirality, or with what random or actually opposite?
So we are testing on random chirality. So what happens? We use the combination of right side and left side, or right and left together. So whether that can plays a role in forming the gel and microdroplets or not. So that is actually the purpose I'm trying to show that is that the screening to find out which chemicals can form the polyester and everything is very complex. So that is the reason that's going to be the ongoing project and process as well. Different teams will do some finding exploration work. Yeah.
So basically, you are telling us that you don't have so any tendency for the right chirality
No.
to go towards some good result. It's not a common, it's not a possible hypothesis towards a common point between different type of life from different planets, for example, that there could be.
Correct.
No statistical
Correct.
anomaly in that sense.
Yeah.
Okay.
That was it.
Okay. Back there.
Just a question I just thought of really randomly. Have you ever thought about like prions, things like self-replicating proteins and their interaction with the B cells?
I'm not familiar with that. Maybe I should take that.
Prions are just they're like almost like a virus. They're not really viruses. They're self-replicating proteins.
Mm-hmm.
They cause like mad cow-like diseases in humans and stuff. It's the reason why cannibals shouldn't eat brains.
Uh-huh.
But they're very primitive and they're not fully understood in that they're self-replicating proteins. So
Okay.
maybe with the new AI stuff like AlphaFold, they can figure it out. But I was thinking that maybe some interaction between B cells with their own self-contained environment. You're talking about for the memory structures, and some types of maybe self-replicating proteins can form the basis of some constant metabolism in biology for some self-replication. Maybe if the right combination occurs, no matter what comets they might come from, maybe it could be unique to the formation of life on this planet, versus maybe it could be partly from Mars, or maybe partly from Europa.
Yeah.
I was thinking, could you ever consider those other types of biological structures, or a compound, like simple ones? They're
So,
not technically
that's-
alive.
Yeah, so that one is talking about the replication of protein itself, right? So for that to happen, it has to have some kind of, a place, right?
Yeah.
So I am talking about that place. So whether it can give that place or not. So that is what the droplet is going to be. Yeah. So the one you are talking about could be a next phase or the phase before the droplet. So it's already could be forming there, and then once the droplet is there, it can enter to it and then start to form like a protocell and everything. So that's why it's also going to be ongoing, and it's a very big thing to find out. It's like a very, very complex topic actually. Yeah.
All right.
Yeah.
Last question, quickly.
So this is going to be a quick question about just a general panspermia theory. So Hayabusa 2 sample, JAXA recently published a finding that Hayabusa 2 sample contains organic matter.
Okay.
I would like to know what's your opinion about it, and do you think it will support, because we don't know yet if panspermia is true or not.
Okay.
So do you think it will support the panspermia theory to be getting closer to the correct one, or any other opinion about that?
Yeah, that's actually interesting because we know that organic compounds were found elsewhere, right? So that means they are something outside of planet Earth. So maybe beyond that, it could have some kind of bigger structure or smaller structure. We don't know that. So that gives the evidence that organic compounds contains in outer space as well. So it's already there. It was delivered to Earth. So that's actually a positive thing, right? It's like maybe that organic compound could have delivered to Earth, or maybe in previous time when Earth was forming, from Earth it has been sent there. We don't know that. So that actually can explain the Panspermia hypothesis as well. So yeah.
So at this point, we just don't know how. We know it's out there, but we just don't know how
Correct.
yet.
Yeah.
Okay, thank you.
Yeah.
Great. Thank you everyone for questions, and thank you. And So Mahendra's gonna stay for maybe half an hour, so feel free to talk to him with your questions and comments.
So I just forgot one important thing, that not important thing, just a random thing. So I'm very sure you have seen the Interstellar movie, right? So in that movie they will be talking about the Panspermia hypothesis as well. As you can, if I mention the name properly, Cooper, the main character, will go to another planet, right? So the space shuttle, the space robot, whatever, it brings fertile organisms. I mean, how to say? Embryo. I don't know what's in biology term, to plant in other planet, right? So they are talking complex molecule from Earth, bringing to elsewhere, and then settle down there. So that's actually a Panspermia hypothesis as well. So by using life as you know it. So yeah, so that was I'm talking about. So that even that movie really can be related to Panspermia hypothesis. Yeah. So that was just simply my comment over here.
Thank you.
Sorry. That's all my extra slides and rubbish.